Good morning, children. Saira, welcome to this session. We're going to continue with the lesson Cell Biology and Biotechnology. We already started with the lesson and we have learned some parts of it. We have learned what is meant by cytology, what are stem cells we learned, and how it is used we learned, and we learned about organ donation, and we started with biotechnology in the second part, and we learned about the benefits of biotechnology. Under that we learned commercial uses, we learned crop technology in that. We learned about the GM crops, okay, and we learned about the hybrid seeds, and we learned about BT cotton, BT brinjal, then golden rice, and herbicide tolerant plants we learned, okay. All these we learned, and biofertilizers also we learned under crop tech biotechnology, okay. Now under next we learned was animal husbandry. How biotechnology is useful for animal husbandry we learned. That is two methods: artificial insemination and embryo transfer. Okay, are the two methods with the help of biotechnology we can use this method, and we can get good quality as well as quantity of milk products or any product of animal. Then we learned about human health, and we learned before the onset of any disease we come to know like disease of heart disease, and even disease like AIDS, okay, diabetes. All these we can we come to know with the help of biotechnology before the onset of disease. Before the symptoms arise, we can come to know about the diseases with the help of biotechnology. And we learned about the hormones like insulin is used for treatment of diabetes, but before it was made with the help of it was collected from the pancreas of horses, but now it is made in the laboratory, and with the help of bacteria it is made. We have learnt it in the previous microbiology lesson, okay. And under vaccines and vaccination also, now we learned that artificially this antigen and making the antigen is the better method, because using the pathogen of half killed or totally killed pathogens was harmful. So this method of taking scientists produce the antigen in the laboratory with the help of the gene isolated from the pathogen and used as vaccine. This is the better method. And we learned about the edible vaccine, okay, which is called as the transgenic potato, which will act against the Vibrio cholerae as well as E. coli diseases caused due to it. And this disease caused by E. coli and the disease against cholera, this is very useful for, it has to be eaten raw. We learned about edible vaccine. And about under treatment we learned that production, it is useful for production of hormones like insulin, somatotropin, and blood clotting factors. And one small part we learned was interferon. This is a group small size protein molecule which is useful for treatment of viral disease. We learned so much, okay. Now let us continue, which is the gene therapy. What is meant by gene therapy we will see now, okay. Gene therapy is to treat the genetic disorders in somatic cells, has become possible due to biotechnology. We learned that there is disorders in the somatic cells. We learned there are reproductive cells and the germ cells, which is called as the germ cells. We learned it in second lesson, as the other cells are called as the somatic cells. So any gene therapy is used to treat the genetic disorder in somatic cells, has become possible due to biotechnology. Example, you learn this by heart: phenylketonuria, PKT. What is the full form of PKT? It can come. This is a disease caused with the genetic disorder in somatic cells. Phenylketonuria arises due to genetic changes in which cells? Liver cells. What are the liver cells called? Hepatocytes. Okay, we learnt about blastocytes, oocytes, all those. This is liver cells are called as hepatocytes. So when there is genetic changes in hepatocytes or liver cells, it is called as the disease is called as phenylketonuria. Okay, so learn this name as well as where it is. It has become genetic changes in which cells, that also you have to learn. It has become possible to treat it with gene therapy. There is a therapy called as the gene therapy. This method is called as the somatic cell gene therapy. What is this method called? Somatic cell gene therapy. Okay. All what are you know this? What all the cells except sperm and ova in the body are called as the somatics. Other than the reproductive cells, all other cells in our body are called as the somatic cells. So any disorder in the somatic cells can be treated with the help of the therapy called gene therapy. Understood? Okay, now next comes the cloning. Okay, what is meant by cloning? There is a picture in your textbook also of the sheep. Okay, production of replica of any cell or organ or entire organism is called as cloning. Same to same, there is no difference in DNA sequence or any structure. Even the features, look, everything is the same. Even the DNA sequence or the gene is also the same. Replica of any cell or organ, that can be even of cell, of organ, or even entire organism can also be cloned. First we'll see about reproductive cloning. A clone can be produced by fusion of a nucleus of somatic cell. Listen this carefully. How it is done? Fusion of a nucleus of any cell, that is somatic cell, with the n-nucleated ovum of any body. That means n-nucleated means nucleus is removed, okay, of that ovum. Some nucleus of the somatic cell is fused with the n-nucleated ovum, that is nucleus is removed from the ovum and it is fused with the nucleus of somatic cells. And for this there is no need of any sperms to produce a new organism. Only the nucleus of somatic cell and an n-nucleated ovum of any organism can be fused, and there is no need of any sperm for this production of reproductive cloning. Understood? So what are fused together? Fusion of the nucleus of somatic cell with the n-nucleated ovum of any organism, okay, any body. That means n-nucleated means nucleus is removed from the ovum and it is fused with the nucleus of somatic cells. Understood? Now therapeutic. Therapeutic means any treatment, okay, treatment. Stem cells can be derived from the cell formed in laboratory by the union of this we saw, somatic cell nucleus with the n-nucleated egg cell. Okay, various disease can be treated with the help of these stem cells. So it is formed. It is formed in the laboratory also with the union of somatic cell nucleus with the n-nucleated egg cell, that is the ovum. Okay, so various disease can be treated with the help of these stem cells. Similar to cells, genes can also be cloned and millions of copies of same gene can be produced. I told you this. Okay, even genes can be copied and multiplied. Same gene can be cloned. Okay, copies can be of the genes can be taken, and those can be used as for gene therapy and for other purposes. See, those you are taking, I told you after 12th, when you take this stream, that time you will get into detail. This intense standard only just a starting part of, just they have touched the topics and what is biotechnology, what is cytology, what is gene therapy, just as molecular biology. All these are little bit part of it only, just they are introduce the topics. Okay, if you want to go deep into this topic, then you have to take this branch of science. Okay, then controlling the inheritance of hereditary diseases, continuation of generations, enhancing the specific tendency may become possible due to cloning techniques. However, there is a worldwide opposition of human cloning on various issues. So there is everybody is against, okay, of human cloning. Suppose there is a same human being and same like us, same, there is no change in the other human being, the cloned. So if so that is not possible. People are against, because if one does crime, the other will be punished, or there are many drawbacks if the human beings are cloned and if we get the same human being like the others. Okay, shall we go to the next now? Now let us come to the next part. We are going to see what are the industrial products. It is called as the white biotechnology. We'll see what all industrial products you can get from biotechnology. Already in microbiology lesson we learned we can get alcohol, okay, from sugar molasses we learned. and we learned many other products, yogurt production and molasses. We learned ethanol. All these we learned in biotechnology lesson. What all you can get: cheese, bread, all these. This all comes under biotechnology only. Whereas industrial chemicals can be produced through less expensive process. We learned all those products here now. Even chemicals can be produced. Example, even alcohol production from sugar molasses, okay, with the help of transgenic yeast. Okay, with that we learned about transgenic, that is change in the gene. Okay, transgenic means. So how example is alcohol production from sugar molasses with the help of transgenic yeast is a method of the industrial products which we can get. Okay. Now coming under fifth type point is environment and biotechnology. It has become possible to solve environment related various problems with the help of bio. We learned all these children how to clean the sewage or the oil spillage. All this lesson, this actually both the lessons are linked to each other. Okay. See, we learned all this by microbial treatment of sewage treatment. We learned in that lesson. Same way. So microbial techniques are already in use for treatment of sewage on solid waste. This we have learned. Sewage is rich in organic matter. We all know this. If such sewage is released in natural in natural water bodies like rivers, and organic matter in it gets oxidized with the help of dissolved oxygen. So it is not safe to let the sewage water into natural water bodies. Due to this, level of dissolved oxygen in water decrease. This we know, when we pollute the water, the oxygen level of that water body will reduce, and due to that what will happen, the aquatic animals will suffocate and they will die. Okay, so due to this, the level of dissolved oxygen in water decreases, adversely affecting the aquatic life. As a remedy on this, sewage water, you all know this, you have to treat the sewage water, polluted water, before leaving it. Only after oxidation, see, as a remedy on this, sewage should be released into rivers only after oxidation with the help of microbial technique. So with the help of microbial technique, first we oxidize all the sewage content, the chemicals present in that, and then when it is left into the water bodies, it is much more safer because it will not decrease the oxygen content. Understood? Because if you leave the sewage water into the water bodies, there is dissolved oxygen will decrease and it will affect the aquatic life. Okay, the microbes we learned all detail about microbes in the previous lesson, isn't it? Microbes are useful on large scale while production of compost by treatment on solid waste material. We learned about landfill method, compost, all this we learned in that lesson. Bioremediation, biopesticides, biofertilizers, biosensors, etc. Some are new concept in biotechnology. So biopesticides and biofertilizers is already there. Now we learn about bioremediation. What is meant by bioremediation? Bioremediation means either absorption or destruction of toxic chemicals and harmful pollutant with the help of plant and microorganisms. So what is meant by bioremediation? So you learn this, children, it can come, means either absorption or destruction of toxic chemicals and harmful pollutants with the help of plants and microorganisms. Okay, if plants are used for this purpose, it is called as phytoremediation. Some examples of remediations are follows. You can learn all this. This is little bit of can come for objectives. Okay, so what is meant by remediation? Bioremediation means it is either absorption or destruction of toxic chemicals and harmful pollutant with the help of what? Plants and microorganisms. Plants are used somewhere, even microorganisms are used as absorbent. They will absorb the chemical and harmful substances from the polluted areas. Okay, so when plants are used, it is called as for phytoremediation. When plants, you all know the name for plants is phyto means plants. So example given here, One is the Pseudomonas bacteria. We learned this. Pseudomonas where we learned for polio. We learned in the cleaning of oil content, isn't it? So Pseudomonas bacteria are useful for cleaning the hydrocarbon and oil pollutants from soil and water. This already we learned. Examples you learn. One is the Pseudomonas bacteria. Okay. And next one, the fern Teres vitata. Okay. The fern, what is the name of the fern? Teres vitata can absorb the arsenic from the soil. This arsenic is very harmful. That can be absorbed with the fern. This is a plant. Okay. Teres vitata. And genetically modified variety of Indian mustard. Okay. A mustard plant can absorb selenium from soil. All these substances, elements are harmful. And sunflower can absorb uranium and arsenic. This all can come for objectives, you better learn. Okay. So for the fern Teres vitata can absorb arsenic. Then the genetically modified variety of mustard can absorb selenium, and sunflower can absorb uranium and arsenic. This is very important. And the bacterium Deinococcus radiodurans is highly radiation resistant organism. It's a highly radiant resistant organism. It has been genetically modified and used to absorb the radiation from radioactive debris. The nuclear debris are all radioactive, okay, from the nuclear power plant. So this bacteria can be used to, because it is radiation resistant organism, it can absorb the radiation from the radioactive waste or the debris. Okay. Then the grasses like alfalfa, clover and rye are used in phytoremediation. So these examples I've highlighted. You learn the examples. This is very important. Okay. Till here we have learned, children. So go through this. We have learned examples of this. Then we will start with DNA fingerprinting from the next video. So what all we learned today, children? We learned about what all we started with the gene therapy. Okay. And we learned about cloning. Okay. Under that we learned reproductive cloning as well as therapeutic cloning. And we learned about the industrial products. We learned how alcohol production from sugar molasses. Then we learned about environment. It is used for cleaning, okay, for removing the oil content from this, and we learned like for cleaning of sewage water, and we learned about bioremediation. What is meant by bioremediation we learned, and that is how we can when plants are used, it is called as bioremediation. Means it is what is bioremediation means? It is either the absorption or destruction of toxic chemicals and harmful pollutants with the help of plants or microorganisms. And we learnt about the fern Teres vitata can absorb the arsenic from the soil, and the genetically modified variety of Indian mustard can absorb selenium from soil. And we learned that a sunflower can absorb uranium and arsenic from the soil. And the bacterium Deinococcus radiourans is highly radiation resistant organism. It can be used to absorb the radiation from radioactive debris. And the grasses like alfalfa, clover, rye are used as phytoremediation. So you learn the example alfalfa, clover, rye. Okay, this can come from for objectives for your board exam, because last year all these have not, it does not come. Okay, children, hope you all understood till here. We will continue the lesson in the next presentation. Thank you, children. Take care. Sai Ram.